So let's continue our reading of this review paper. So according to our colleague here, we were reading page 168 and we had finished we were in the last paragraph during the translation from activation to repression. So maybe we can start today from the right. Please, let's start. Kokone.
During the transition from activation to repression, the clock BMAL1 complex interacts with the per-cry repressor complex at the beginning of the night as the levels of per and cry accumulate and translocate to the nucleus. The per complex is large and contains at least 25 proteins. Initially, clock and BMAL1 are associated with two components of the MI2 nucleosome remodeling and the acetylase. Transcriptional co-replacement complex, CHD4, chromodomain helicase DNA binding protein 4, and MTA2, metastasis associated 1 family member 2. Interestingly, per complexes without clock BMAL1 like CHD4 and MTA2 will contain other members of the NURD complex, such as MBD2, meso CpG binding domain protein 2, GATA-D2A, GATA zinc finger domain containing protein 2A, HGAC1, and RBA-P48, retinoblastoma binding protein P48. This finding indicates that during this transition phase, there is a split NURD complex that then comes together to form a reconstituted NURD co-repressor complex to inhibit clock BMAL1 transcription. The clock BMAL1 complex also recruits polycomb repressor complex 2, leading to the trimethylation of H3K27 at target genes and thereby inhibiting gene expression.
Okay, thank you very much. Let's stop there. There are a lot of names. You don't need specially to remember each of the names. This is not what is important. So, what it simply describes a little bit in a complex way is that there is a transition. As you know, clock and BMAL, as we saw before, it's a transcriptional activator. When it binds to promoter of target genes, it promotes transcription. But then, clock and BMAL complex will interact with the per-cry repressor complex at the beginning of the night. But it's at the same time as the level of per and cry level accumulate and translocate to the nucleus. So it's a little bit funny, right? Because it says that clock and BMAL complex interact with per-cry repressor complex when per and cry accumulate. Why is it strange? Do you understand why there is something that's a little bit strange? It's correct, but it's really strange in the sentence. What they say is, here I am drawing the theoretical curve of per expression. And what they are saying is that at about this time, if you look in the promoter of clock and BMAL target gene, this is clock, this is BMAL, per and cry complex will associate. But it's very strange, right? Because if they associate with clock and BMAL, why is the expression of clock of cry and per increase. Right? Or not? So this is the first sentence. During the transition from activation to repression, the clock-BMAL complex interacts with the per-cry repressor complex at the beginning of the night as the level of per and cry accumulate and translocate to the nucleus.
Maybe there's a delay in effect.
Maybe because there is a delay. So when per and cry interact with clock-BMAL, the inhibition is not immediate. Because otherwise, as soon as per and cry interact with clock-BMAL, what we should have is an immediate decrease. But we don't have that. In the beginning of the night, when these interact together, still the level of per and cry increase, and only later the repression comes. So this is what they explain. Something else is happening. When per and cry binds to clock and BMAL, there is a whole modification of the chromatin at that location that eventually lead to repression. And it might be a few hours between the binding of per and cry to clock and BMAL and the actual repression of transcription. It's not immediate. There is a lot of remodeling happening at this location. And as they say, the per complex is large and contains at least 25 proteins. Most of these have not been identified. So it's not only per cry complex, it's per cry complex and then X, Y, Z, many other proteins we don't know. So it's a very big complex, very tightly regulated in a complex way. Still, there are many things we do not understand. And there are many names, as you read, but the key words here, for example, deacetylase, for example, helicase, and all these proteins, they will collaborate to essentially shift from promotion of transcription to inhibition of transcription. Why deacetylase? Why would you have MI2 nucleosome remodeling and deacetylase NURD, transcriptional co-repressor complex? What I am attracting your attention on is why deacetylase, why it is important, this deacetylase.
Because the acetylation is make DNA easier to access and is promote the transcription. So
yeah, acetylation is something that it depends where, but acetylation of histone protein is something that promotes transcription. So if you have a shift at some point, a shift from activation to repression, then something must remove this acetylation. So this is why in this complex there are deacetylase that will remove the acetyl from the histone and shut down clock-BMAL mediated transcription. So essentially this is what this complex paragraph means. You have another one there at the end of the sentence in the middle of the last paragraph. So there is HDAC1. It's also deacetylase. So on the page 169, interestingly, per complexes without clock-BMAL lack CHD4 and MTA2, but contains other member of the complex and then blah blah blah. HDAC and RBAP48. So HDAC1, it's also histone deacetylase. This is also one of the same activity. Then the last sentence that you read, the clock-BMAL1 complex also recruits polycomb repressive complex 2, whatever the name, leading to the trimethylation of H3K27 at target gene and thereby inhibiting gene expression. What is H3K27?
27 is lysine on the third histone.
Yeah, so K is lysine. 27 is the lysine number 27 in the protein. And H3, this is the histone H3. And so what it means, if you add 3 methyl on the lysine 27 of histone tail 3, then it also contributes to the inhibition of gene expression. All right. So then,
I have
Yeah.
a question. So it seems like you're talking about the transition phase. Basically, the per-peptide complex, they can inhibit the transcription of the target genes of the BMAL1 clock. But I mean, in this paragraph, there's like it seems that they're saying, like on page 169, before the last sentence I read, it says it also inhibits clock BMAL1 transcription. So is it also directly inhibiting the transcription of clock and BMAL1?
no, this is well spotted. I think they forgot clock BMAL mediated transcription.
Okay. Yeah, that's why.
Yes, it is very, I agree. So it is not the transcription of clock and BMAL1 mRNA. It's the clock BMAL mediated transcription. Okay. So if everything is okay, let's continue reading. So you can go forward now.
In addition, clock BMAL1 recruits the DNA damage binding protein 1, culin 4, ubiquitin ligase complex to circadian target genes to promote H2B monoubiquitination at E-box sites to covalently mark genes for subsequent recruitment of the PER complex. At the peak of repression during the middle of the night, the PER complex is also associated with the transcriptional repressor complexes SIN3 HDAC and HB1 gamma SUV39H histone methyltransferase, which contribute to clock BMAL1 repression through the deacetylation of histone H3K9 and H4K5, as well as demethylation and trimethylation of H3K9. The PER repressor complex also includes NONO, a protein involved in RNA processing, and WDR5, a component of a histone methyltransferase complex. At later time during the repression phase, PER complexes include the RNA helicases DDX5 and DHX9, as well as the large subunit of RNA polymerase II and ACTX, a helicase that promotes transcriptional termination. Thus, the recruitment of several chromatin-modifying complexes as co-repressors is necessary for PER-mediated and probably CRY-mediated repression of clock BMAL1. During the circadian cycle, transcriptional activation and repression are accompanied by dynamic epigenetic transitions in chromatin state that poise the genome for rhythmic gene expression.
Yes, nicely read. Thank you very much. So this is more of the same. I would say the interesting bits that we see here is maybe something that we have not seen before. ubiquitin ligase complex to circadian target genes to promote H2B, so histone 2B, monoubiquitinylation at EBOX sites to covalently mark genes for subsequent recruitment of the PER complex. That's a little bit funny. How do you know ubiquitin? In which context have you learned about ubiquitin? Have you learned about ubiquitin in the past?
Degradation.
Degradation. Degradation of what?
Protein.
Degradation of protein, yes. What is ubiquitin? What kind of molecule it is? What kind of molecule it is? Is it a sugar? Is it a fatty acid? Is it just organic molecule like some carbon? What is ubiquitin? Is a small peptide of a specific sequence, and indeed ubiquitin is normally something that is added to protein, and it's like a label. Please destroy this protein. But in some cases, especially the ubiquitination of histone, it's a mark that invites the recruitment of protein. So in this case, what they found is that when H3B is monoubiquitinated, then what you get is it promotes the recruitment of PER proteins to further increase this transcriptional repression. And something that is important is monoubiquitin. So this means only one ubiquitin. When you have degradation of protein, you have many copies of ubiquitin added after each other. So this is a little bit different. It is a little bit different. Associate with the transcriptional repressor complex, which contribute to clock-BMAL repression through the deacetylation of histone H3K9. So again, not only one site is deacetylated, many sites in histone is deacetylated, all together promoting repression. But then something that is a little bit interesting is that it is easy to understand why the binding of PER and CRY would lead to all these things that stop transcription. But what you can see is that the PER repressor complex also includes NONO, a protein involved in RNA processing. And then at later times during the repression phase, PER complexes include the RNA helicase DDX5. And as well, the large subunit of RNAP2 and SETX, a helicase that promote transcriptional termination. Helicase? What is a helicase? What is a helicase? This is kind of interesting. And so the first helicase we see, this is something that is associated with DDX5, right? It's RNA helicase. What is a helicase?
Use the reading of DNA to create the RNA strand.
Yeah, so helicases are needed for regulation of transcription, but Yes. What else? This is not DNA helicase. This is RNA helicase. I mean, helicase, the name of this enzyme means somehow something that makes helix, right? It's an enzyme that promotes helix formation, or that sort of changes the state of the helix. But it's easy to understand if it's about DNA, but this is RNA helicase. So can you guess what might be the function? Why would helicase be required? Why would they be required? I think it's easier if you have in your mind what really happens at the level of transcription. So what you have here is the DNA template of the target gene. And then what you have here is you have clock and BMAL that sort of promote transcription, right? And transcription means that not only clock BMAL, but somehow the recruitment of a big complex of protein that actually make a copy of the DNA. This is RNA. This is RNA, right? And as the polymerase starts to actually be recruited to this location, and many, many times, there will be not only one polymerase. One polymerase will bind and will start transcription. Then another will start and start transcription. And they will all release RNA. But the RNA will become, as this polymerase advances to the 3' end, this RNA will become progressively longer, right? Until it's finally made. But the RNA should not be new, okay? The RNA should not be left hanging like this. A lot of protein will also interact with RNA to make sure that the RNA is protected, packaged, and has the right structure to continue all this process. So this is why not only you have proteins that are specific for the initiation of transcription in itself, but you also have proteins that are required for RNA protection. And if RNA has a specific structure, it will also promote the termination of the transcription. So it's not only about doing something with the DNA, but it is also something that has to do with changing the RNA that is being made, changing the structure of the RNA being made. And still at the moment, we know a lot of things about how clock genes are regulated at the transcription level, but something that is very hot now in circadian research is what happens at the level of DNA. At the level of RNA, sorry. How is RNA processed? And we will see that later. So we have the first suggestion here that all this complex also contains proteins that do not really do anything with DNA, but they do something with RNA. And that is also important for the clock. Okay. That is also important for the function of the clock. And I should maybe mention here that in textbooks that you buy, you have a very step-by-step idea of having your DNA. Maybe I've said that before, but I want to make sure. Then, from this DNA, you have pre-mRNA. Is the pre-major messenger. And this pre-mRNA, so this is transcription. Then here you have mRNA with the cap and the poly-A tail. What is this one? What is this step?
Translation. Oh,
No.
Post.
It's post-transcription, but translation is also post-transcription.
Post-transcription.
What is this one?
Splicing.
Yeah?
Splicing.
Splicing, yes, for example. Splicing, or more generally, RNA processing. So not only you need to splice, but you need to process RNA. You need to add the poly-A tail, because the poly-A tail is not encoded into the DNA. It's added afterwards onto the RNA. So all these steps, in books usually they are step by step. First you transcribe, then you splice, then you add RNA. This is actually not true. Everything happens co-transcriptionally. Even the cap, which is usually added later in textbook, the cap is added only after 20 or 25 nucleotides go out of the RNA polymerase. The cap is added very quickly. And splicing occurs in the same time of transcription, because RNA polymerase contains different domains that recruit splicing factors. So everything happens at the same time. And it's extremely dynamic, and there are many things we still do not understand how it is regulated. But it seems that whatever happens, the clock is there and regulates each of these processes. Okay? Any comments or questions or... So we see histone methyltransferase, same as acetylase or deacetylase. Okay. Adding methyl to some lysine of histone will promote transcriptional arrest.
So what does the RNA helicase do?
So RNA helicase, so there are different families of RNA helicase. Some of them are actually packaging the RNA like, as I said briefly, RNA should not just be like a noodle, just hanging there. It should be associated with protein. But to associate with protein, it should form secondary structure, like, for example, things like that. These are called hairpins. And it's secondary structure in messenger RNA. And there are helicases that sort of promote formation of this kind of structure. They stabilize RNA. But some helicases, they associate with these and they stop transcription. So in this case, the helicase that they are mentioning is helicases that have been involved in stopping transcription. Probably they sort of interact here and they stabilize RNA in such a way that it interrupts further transcription. Exactly what happens at the molecular level, what is the structure of RNA, we don't know. But it seems to be helicases that interact with RNA and changes the secondary structure to prevent elongation of the RNA. Okay. So the whole synthesis of this paragraph is really during the circadian cycle, transcriptional activation and repression are accompanied by dynamic epigenic transitions in chromatin state that poise the genome for rhythmic expression. This is really the synthesis of that local paragraph. Let's continue. I'll begin something.
Regulation of circadian period length by repressor stability. The stability of the core repressor proteins is an important determining factor in the dynamics of the transcriptional feedback loop, which in turn regulates the period length of the oscillation. The stability of PER and CRY is regulated by their phosphorylation state and by their ubiquitination by E3 ligase complexes. For example, the TOL mutation in CSNK1E, which encodes casein kinase 1 epsilon, shortens the circadian period length in vivo. Moreover, this mutation causes the destabilization of PER proteins by phosphorylating a regulatory site that marks the PER proteins for ubiquitination by beta-TRCP, also known as F-box WD repeating containing protein 1A, and proteasomal degradation. By contrast, mutation in the gene encoding the F-box protein FBXL3, which is one of 4 subunits of the SCF E3 ligase complex, lengthen the circadian period and stabilize the CRY protein by decreasing ubiquitination and subsequent proteasomal degradation. In addition, CRY ubiquitination by SCF is triggered by 5' AMP activated protein kinase mediated by. In addition, CRY ubiquitination by SCF is triggered by AMPK mediated phosphorylation, which provide a link to metabolism via nutrient responsive AMPK activity.
Okay, thank you very much. So we are back with casein kinase and the level regulation of the clock by regulating the stability of these transcriptional activator and transcriptional repressor. So mainly it's discussing about the stability of PER and CRY. Because PER and CRY are the negative complex, regulating their stability has a very strong impact. If they are very stable, it means that the repression will be very long. And if they are very quickly degraded, then the repression will be very short. So then there is no surprise that there are mechanisms that regulate their stability. But in the text, there is something that is a little bit, maybe not funny, but a little bit difficult to understand. Maybe it's okay, but so about FBXL3, so this is by contrast, mutations in the gene encoding the F-box protein FBXL3, which is one of the 4 subunits of the E3 ligase complex, lengthen the circadian period and stabilize CRY protein by decreasing ubiquitination and subsequent degradation. So that's easy to understand, right? You make a mutation in ubiquitin ligase, it's a protein that adds ubiquitin on a different protein. So if you make a mutation in a ubiquitin ligase that ubiquitinates CRY, then this mutation will decrease ubiquitination of CRY protein. So cryoprotein will be more stable because it's not ubiquitinated anymore. But then what they say about casein kinase epsilon is different. They say the tau mutation in casein kinase epsilon shortens the circadian period length in vivo. So, as opposed to the FBXL3 mutation, the FBXL3 mutation stabilizes cryoprotein and the period becomes longer. Okay, that's kind of you understand intuitively. Okay, but with this casein kinase epsilon, it's a tau mutation that shortens the circadian period because this mutation causes the destabilization of PER protein by phosphorylating a regulatory site that mark PER protein for, where was it? That mark PER protein for ubiquitination by beta TSCP. So you have a mutation in casein kinase epsilon that causes the phosphorylation of PER protein. Why is it funny? I find it's funny. FBXL3, you have a mutation that decreases ubiquitination. That's easy to understand. The mutation is sort of destroyed, FBXL3. But for CK1 epsilon, as we saw before, it makes a short period. But they say it's a short period because it caused the phosphorylation of PER. So, if you think about the mutation in CK1 epsilon and the mutation in FBXL3, what might be different? What might be different? Maybe it's a little bit. What might be the difference between these mutations?
So the CK1 epsilon binding, it's a kinase, so it's supposed to phosphorylate something, but the mutation, like in the mutation, it doesn't change its ability to phosphorylate, it still phosphorylates. But in the other case, I mean, if you have a mutation, it no longer ubiquitinates. Like, I don't know whether I've explained this. Like, one is locked, like the FBXL3, it loses its function of ubiquitination, but CK1 epsilon kinase is still phosphorylates.
Yeah, so this is exactly what they don't explain that at all because they sort of think it's, you know, but if you think about it, it's a little bit difficult. So you have Cry1, protein that first gets ubiquitinated by a protein called FBXL3. And then once it's ubiquitinated, then Cry1 sort of falls apart. But if you make a mutation in FBXL3, what they say is that there is no more ubiquitin added on Cry protein. So Cry protein is not degraded. And this mutation, because this stabilizes Cry1, then this means that the period, which I write as tau, increased. That's easy to understand because Cry are stable for longer. Now, casein kinase epsilon is very strange because it's a bit similar. We have PER protein, which are phosphorylated by CK1 epsilon and phosphorylation by CK1 epsilon cause per protein that are phosphorylated, they become ubiquitinated, and then they are broken down. That's the mechanism. That's what they say, right? But what they say is that the tau mutation sort of cause the phosphorylation of per that cause degradation. That's what they say. Does it make sense? This is very easy. But what they say is that CK1 epsilon phosphorylate per, which then cause ubiquitination of per protein and to their degradation. But when you make the tau mutation, actually, if you it's different from this one. The tau does not become longer, the tau becomes shorter. Because somehow you promote phosphorylation of per that leads to degradation. So we need to make some theories there. Because this delta FBXL3 mutation and this tau mutation, they probably have very different effects. And actually, this is not really understood yet. So what could be the possibility? What you say was true. So it's a mutation, but the mutation may not really affect the kinase ability of CK1 epsilon. So what could be affected? What could the tau mutation do? Because they say phosphorylation of per leads to ubiquitination, leads to degradation, but the tau mutation leads to phosphorylation that causes degradation. So it's a little bit confusing. So what do you think, what is hiding behind there? So I'm asking you there to think hard about what is hiding behind.
Site where it phosphorylates.
That's nice. The site where it phosphorylates. Yes, indeed. Maybe you have the per protein, and phosphorylating the per protein here may cause ubiquitination and degradation, but the site here may cause quicker destabilization. That's very possible. That's one possibility. What else? So maybe per proteins have more than one site they can be phosphorylated. That's one possibility. What else? And then according to, if we assume this is true, if per really has more than one site in which they can be phosphorylated, then what will be the effect of this mutation? Because we have to relate to the mutation, right?
If the proposed theory is true, then there are two phosphorylation sites. The one for beta TRCB binding site. The one.
Yeah, continue.
The one for beta. Occasionally, the two phosphorylation sites are equally phosphorylated. But for example, in this mutation, the one for TRCB binding site is more likely to be chosen.
Yes, That's maybe very true. Is that maybe the mutation, the tau mutation, may affect the ability of casein kinase epsilon to phosphorylate per protein on one of these sites, but the other one is not affected. Or it may promote phosphorylation of one site and the other one is not affected. So that's very true. So maybe in this case, what happens is not like maybe, what happens is that the mutation of CK1 epsilon promotes, I want to check something here. Yeah, so what the mutation could do may not involve two phosphorylation sites, but simply this is a loss of function. This is a loss of function mutation that will completely blunt the activity of FBXL3, but while the tau mutation may be more active at phosphorylating this site. So we don't necessarily need to have two sites. We may need to have only one site, but somehow this kinase is more active. And this is called a gain of function mutation. Gain of function. A gain of function mutation. So somehow the casein kinase 1 epsilon tau is more active than the one time. That's very possible. That's very possible. That's what actually people think at the moment, that it's a gain of function mutation.
So the casein kinase cannot function at the FASBD site anymore, and it can only function at the beta TRCB site.
Yes, do they? I'm kind of confused. What do they say about the fast So this mutation causes the destabilization of per protein. Yeah, so they do not, in the text, they do not mention any other sites, right? They just say in this text, the tau mutation shortened the circadian period length in vivo. Moreover, this mutation caused the destabilization of per protein by phosphorylating a regulatory site. You're already reading ahead. Okay, so then let's read ahead. We have more information.
The stability and turnover of paired proteins is primarily regulated by several mechanisms, including by CK1 delta and CK1 epsilon. By the balance between these two proteins, kinases, and phosphatase 1, and by the stoichiometry of paired proteins in these kinases, recent work has shown that a dual kinase, multi-site proton switch, regulates pair stability and its ubiquitination by the beta-TRCP E3 ubiquitin ligase complex.
You can continue, it was very short.
Okay. And for cryoproteins, their stability and turnover are regulated by the balance of the two paralogs FBXL3 and FBXL21. Contrary to expectation, FBXL21 antagonizes FBXL3 to reduce the E3 ligase activity of SCF on Cry1 and Cry2 to stabilize them. Although SCF does possess E3 ligase activity, it is less efficient than SCF in the ubiquitination of Cry. However, FBXL21 interacts more strongly with Cry than FBXL3, which protects Cry from SCF activity. The subcellular localization of FBXL3 and FBXL21 also differ, with FBXL3 acting as the primary E3 ligase for Cry in the nucleus, whereas FBXL21 acts as the primary E3 ligase for Cry in the cytoplasm. Finally, in genetic experiments exploring the interaction of the CK1 epsilon tau mutant with the FBXL3 mutant, A broad range of periodicities was seen depending on the combination of alleles, indicating that the PER and Cry pathways independently affect the ultimate period length in vivo.
Okay, thank you very much. Yes. So we have more information now on the casein kinase 1 delta and epsilon. So what is the current accepted mechanism? What is the current accepted mechanism? Is that, as you said, and the PER protein has actually, more than two, but has actually two sites of different function. One site, let's name it FASPs, and this site, let's name it beta TRCP. When the beta TRCP site is phosphorylated by casein kinase 1 delta or epsilon, this causes the degradation of PER protein by the ubiquitin ligase and further degradation. But the FASPs or FASPD, whichever site, phosphorylation at this site will not lead to PER degradation, but will stabilize PER. This leads to stabilization. This leads to degradation. This leads to degradation. So what is thought is that the mutant CK1E tau has lost the ability to phosphorylate PER on the FASP site. And so PER2 is not stabilized. In the same time, CK1 epsilon may still have or higher activity of phosphorylation at the beta TSCP site. So even though it's a mutation, it will cause quicker degradation of PER. And we can see with Cry there is something very similar, but here a kinase apparently is not involved, in which we have two different ubiquitin ligases, FBXL3 and FBXL21. They have slightly different function. One protects Cry, the other one degrades Cry. So the conclusion of that is that the clock is extremely complex. Okay, and you should not see like this works in all tissue the same way. If you check the cell in the brain and if you check your cells in the liver, exactly the same thing will happen. Not at all. As they explain, the expression of all these proteins that regulate the clock will be different. So the clock will be regulated in a different way in different tissues. Okay. But it seems to be something very general, is that there is always this image like this of the clock. The clock is a balance between something. It's a balance between day and night. It's a balance between stabilization and degradation. It's a balance between nuclei versus cytoplasm. It's a balance of very different things. So it's not a surprise that it's so tightly regulated. There was a very nice, so the FBXL3 and FBXL21 paper, I will show you if I can, it's kind of a nice picture. So, co-workers and me, when we were working in Aberdeen, we actually discovered FBXL21. But a few years, it was not a very important paper, but we say, hey, by the way, FBXL3 exists, but apparently FBXL21 is more expressed than FBXL3, so it might be useful. And then a guy in Japan, which is in Tokyo, a few years ago, he was working on this thing as well, and he published a very, very nice paper about the role between FBXL3 and FBXL21. And the cover of the paper was very funny. The cover of the cell that he published in was very nice. I will show it to you because it's a nice cover. It's just for the fun of it. And I find this picture shows very nicely what happens. While we wait for this machine to sort of become alive, any question on this? So here the phosphor switch, which you have a definition on the left here. In the case of the PER2 protein, a phosphor switch model is proposed whereby two competing phosphorylation sites, one for the FAST-D site and one for the beta-TSCP binding site, determine whether PER2 has a fast or slow degradation. So this is what has been dubbed a phosphor switch. It's a switch of phosphorylation. So the guy who has published this is very nice because it was in Cell paper. I hope I can find. Yeah, this one, Hilano. And they managed to, when you publish a paper, you can ask the journal in which your paper is accepted. We have a cover. We have a proposal for the cover that corresponds to our article. Would you like to accept? And they managed to get it. So this team is a team from Tokyo, and it's a guy called Fukada, Yoshitaka Fukada, who's kind of a nice guy. But whether we can see. So they mention, despite the similarity between these FBXL proteins, the physiological role of FBXL21 in circadian clockwork remains to be elucidated. And then this is a paper which I collaborated with a guy from France. But their study is much more complete. They made knockout of FBXL3 and studied. So it's a very, very nice paper. But I just want to see whether I can get the cover for this guy. If I don't find correctly. Here comes nothing. Yes! Nice. Now I want to see this guy. Okay, I'll just make this thing bigger. Zoom. So you see, this is the cover that they proposed and they got it through. So you have the clock behind, and then you have these two FBXL3 protein who sort of regulate the clock. One is pushing it faster, the other one is pushing it. So this is the image, and this is something that is pervasive in clock. There is always two balance of things. So it's a kind of very, very nice. And it's just for the fun of it. Okay. I'm going to read the next paragraph. We still have time. Genome-wide clock architecture. Although previous work has examined the transcriptional regulation of clock BMAL1 target genes, such as DBP, PER1, and NR1D1 in detail, the identity of the circadian systrome, definition can be found on the left, has only been recently defined, using chromatin immunoprecipitation followed by sequencing, ChIP-seq. Several research groups have identified genome-wide DNA binding sites for BMAL1, clock, NPAS2, PER1, PER2, CRY1, and CRY2. I think this paragraph deserves explanation before going further. Especially chromatin immunoprecipitation followed by sequencing. What the hell are they speaking about? Have you come across something similar to the ChIP-seq? First of all, sequencing. Sequencing, maybe you should know what is sequencing. Sequencing is simply to analyze what is the sequence of DNA that you have or the sequence of RNA that you have. Okay? There is a guy 20, 30 years ago called Sanger. There is a Sanger Institute, you know, the Human Genome Project and everything. This guy has invented the method to sequence whatever bits of DNA or RNA you have. But in this case, it's much more complicated than that. What they are saying, it's next generation sequencing. And to understand what's coming, I think you should understand what is this next generation sequencing. What I am drawing on the board, imagine I am drawing it about 22,000 times. But I don't have time to write something 22,000 times. I will write it only for one gene. And there is about 22,000 genes in human. Okay? So for each gene, you have messenger RNA. And not only that, but you may also have different transcripts coming from the same gene, alternative splicing. What sequencing is, not in detail, is not only to measure not only to detect what is the sequence of each of these transcripts, but to count how many copies of it there is. So at the end, you know, okay, this messenger RNA, there are 5,000 copies. This messenger RNA, there are 300 copies. This RNA, there is 500, 200, 5,200. This RNA, there are 10,000 copies. And you can do that for every single messenger RNA that you have in your cell. At the same time, you don't need to measure separately. You extract RNA from your cell, and in one single experiment, you know how many copies of everything you have. This requires strong statistical skills. Well, it requires to use a computer that has strong statistical skills, okay, because this takes a long time. So this is what is seq, the seq part. You can quantify and analyze the sequence of what you have in your sample. Now we are going to do ChIP-seq. What I drew here with RNA, you can also do that with DNA. You can also sequence DNA, it's just the same thing. But in the case of ChIP-seq, we are at the level of DNA. And the chip is the key word. This is chromatin immunoprecipitation. In the word immunoprecipitation, what do you understand in the word immunoprecipitation? What do you think it will involve? Immunoprecipitation.
Defined transcription factor of the chromatin.
On a more basic level, what the word immunoprecipitation contains? Immuno, what does it mean, immuno?
Using antibody.
Exactly. Immuno, this is related to antibody. Precipitation is just like you have a tube and you pull things at the bottom. And what you want has been precipitated at the bottom. And at the top, you can trash it. Okay? And immunoprecipitation means that you pull it down using antibodies. You pull it down, you precipitate using antibodies. Okay? So now we understand each other. You know now that clock, BMAL1, per, cry, they bind to DNA and to each other. So if you imagine that at different times during the day, you may find clock and BMAL1 bound on DNA in different locations in the genome. So for example, here, this is the gene that codes for per protein, for example. So you know that at certain time, clock and BMAL will be bound, right? Okay. So what you do, if you want to know where BMAL1 is located at different times during the day in the genome, then you use an antibody that will recognize BMAL1. It has to be a good antibody. But before you do that, you must cut DNA. You must cut DNA so that the DNA here is cut. But the DNA that is bound to BMAL is kind of protected from being cut. So when you cut DNA, what you have is something like this. You have a sequence of DNA that is bound to a big BMAL complex that is protected from being cut. So in your samples, after cutting, so please forget the antibody for now. After being cut, you have a lot of genomic fragment, genomic DNA fragment. Some of them are not bound to clock BMAL. Some of them are bound to clock BMAL. Many, 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 many. Then these fragments, you mix them with an anti-BMAL antibody. That will then bind to the BMAL1 you have. Of course, it will bind to BMAL1 that is free. But that you don't care so much. But then the precipitation part of this is the immuno part, okay? The precipitation part is simply you are binding this antibody to very big beads. These beads can be magnetic, Jicky beads, or they can be heavy, made of some plastic. And what you can do, if you put all this mix in a tube by centrifugation, or if you let's say you are using magnetic bead, it's easier to understand. If you are using magnetic beads, then what you can do is, although it's a little bit cartoonish, let's do like this. You know, this is an old-fashioned magnet, right? If you put the magnet here, then all these B-mal complex antibody beads will go at the bottom. And then you remove the top, and then you take what you have at the bottom, you remove the antibody, you remove the beads, you remove proteins, and you sequence and quantify each bit of DNA that you have. And not only you can identify the sequence of these B-mal-bound fragments, but you can quantify them. How can you quantify? Imagine I do this experiment in the liver of mouse, because you need quite a lot of sample. And I do this experiment in the morning at 8 o'clock, then at midday, then at 4 o'clock, 8 o'clock, 12 o'clock, 4 o'clock, 8 o'clock, for example. Every 4 hours I do the same thing. And I want to know where and how strong B-mal binds to DNA at each of these time points. So you understand, it's easy to understand how you might be able to know where it binds, because you know the sequence. So just because the sequence of the genome is known, if you have a fragment of a specific sequence, it's easy to localize in the genome. But how can you quantify how strong the binding of BMR1 is, or CRY, doesn't matter? How can you quantify, how can I tell people that if I do that, I see that in the liver, maybe at 8 o'clock in the evening, BMR is strongly bound compared to other time points. How can I say that? How can you quantify?
Maybe you pick a control that is uniformly expressed throughout the day, and we make a ratio between the number of copies you have, like whatever the genes you're interested in, with that uniformly expressed gene.
Yeah, you need to have a control sample in which you have not bound DNA. You need to have a control. But yeah, so you need to have a control, for example, where you use an antibody that is not against BMR1 to sort of remove the noise you have from this data. But how can you quantify that? If you remember, BMR1 has a rhythm of expression, right? So you kind of imagine that at this time point there is a lot of BMR1. So it might be bound very strongly because there is a lot of it. But this binding strength, where will it come from? Where will it come from? From your samples. The sequence, you know, it's easy to understand how you can get the sequence, but because, I mean, what I explained to you before was about RNA. So this is easy, right? You have one gene that is producing many RNA, so it's easy to understand that some mRNA you will detect 100 copies or 1,000 copies. But DNA, you have one copy of gene, 2 copies of gene per cells. So how can you quantify how strongly B-mile is bound?
How do you remove the transcription factor from the genes? In the previous step, you have to remove the transcription factor on it. So can we do something like that step
to remove the transcription factor from the gene? What do you mean, remove the transcription factor from the gene?
So before sequencing DNA, you have to remove everything?
Yes, before sequencing, you need to get rid of protein. You need to have pure DNA.
Yes, and how do you get rid of the protein?
You get rid of the protein usually by using protease. So using enzymes that will degrade protein.
So to quantify the strength of the binding, maybe we can do it many times. And
you don't have money to do it many times. You don't have money to do it many times. You are looking too complicated. You are looking too complicated. It's true that in one single cell, you may have many different copies of each messenger RNA, and you have only 2 copies of genes. But then, what you are doing here is certainly not with one single cell.
I know with multiple cells, would you have to use different mice then, because you'd be removing the liver every 4 hours?
Oh yeah, of course. Yes, yes.
So how do you know it's standardized, like that? How do you know that one mouse doesn't naturally produce more than the other mouse?
You don't. Welcome to biological variations.
Okay.
This is where we need to analyze statistically. There will be variations. Usually, these kind of analysis, you can use 2 or 3 or 4 mice if you have a lot of money, and then you will have variations. And you hope that what you can detect can overcome these variations. But you can limit these variations. For example, if you use mice of exactly the same genetic background, then you have very little variations. So, what about the quantification?
So you use a lot of cells.
Yes, you use a lot of cells. You use a lot of cells. And then maybe, because you have biological variations, if you use a lot of cells, you may find out that at this time point, BMAL is very low, but still you will detect that BMAL1 is there on some part of the DNA. But in not all of your cells, so in the end, when you take many cells, you have one huge piece of the liver, then it might not be significant. But at the time where all these mice have a lot of BMAL1 in their liver, then you will find out that BMAL1 is always there at the same location in all these mice. So when you immunoprecipitate these things, you will have many times the same DNA fragment, but from different cells. So the amount of this DNA fragment is what you quantify. And you can quantify because it's from many different cells. Okay? All right. And then it's almost time. Do we have a figure? Now we don't have a figure. We need to read further. So let's read the next paragraph.
Genomic targets of the core circadian activators. Occupancy of clock BMAL1 at the DBP locus, which encodes a transcription factor that modulates the expression of clock output genes, is circadian, with peak binding occurring during the middle of the day. At the single cell level, clock and BMAL recruitment to E-boxes in DBP are interdependent and circadian. BMAL1 binding is dynamic and stochastic, and fluctuations in binding depend on proteasome, which is consistent with a Kamikaze model of transcriptional activation. On a genome-wide level, clock and BMAL1 occupancy also peak within a narrow window of time during the middle of the day, and the phasing of clock BMAL1 occupancy is not tightly phase-locked with the RNA expression of their target genes. This flexible phase relationship can be partially explained by the combinatorial action of other cis-acting elements, RREs, and D-boxes within the tripartite clock gene network. For example, CRY1 expression, which peaks much later in phase than her genes, is regulated by both E-boxes and RREs.
Okay, thank you very much. So quite complex again. Anything? I think the beginning is okay, right? So this Kamikaze model, it's a little bit too much details, I think, here, but what it means is that it seems if you want a maintained activation of clock BMAL mediated activation, you must always make new BMAL1 protein. You don't have one BMAL1 protein sitting there continuing activating the transcription. It must be replaced all the time. Okay? So you put BMAL1, you activate transcription, you degrade BMAL1, and you put a new BMAL1. So it has to be recycled pretty quickly. Otherwise, transcriptional activation stops. And the tripartite, this is in figure 2, right? This is what they explained in figure 2. So you have many interlocked feedback loops that sort of explain these things. Any question for this part? Maybe that's not so complicated. Everybody's okay? Yeah? All right, then I think we can stop here before continuing next week. If you have any question, please don't hesitate. So next time we will see the figures that are related to what we read, and you will see exactly what happens